Hello, welcome to 13 Universal. This is A-level CI biology. I want to say that every question we're using here is from A-level past questions and math scheme. And we are going to be doing biological molecule series 5. This is a topical revision on biological molecule towards the ongoing May-June 2024 series, okay? Exams. So take your writing material and let's solve some questions, okay? So the table shows the observation recorded. You can see that. Shows the observation recorded from tests for biological molecules on 4 samples. That's samples A, B, C, and D. Which conclusion is correct? Okay. So let's pause the video and then try the conclusions, guys. Right? So if you walk over it, just pause the video and try it. Benedict solution blue. So blue, what does it blue mean? Benedict solution is used to test for reducing sugar. Blue means reducing sugar is absent. Okay. Burette reagent is used to test for protein. Blue means it's absent. Okay. If reducing sugar is present, it would have changed to green or yellow or orange or red. If protein was present here, this would have changed to purple, okay, or mauve, okay. Now iodine solution, alright, changes to blue black. That means starch is present, okay. So this doesn't contain, reducing sugar doesn't contain protein, it contains only starch. So this A is correct. But if you check B, conclusion says contains reducing sugar only. It does not even contain reducing sugar because it's blue. Alright. What is wrong with C? Okay, it contains reducing sugar and protein. Yeah, it contains reducing sugar. It doesn't contain protein because it would have changed to purple. D contains starch and protein. It doesn't contain protein. Right? Protein is blue. And it contains reducing sugar, but it was never mentioned. Okay, so A makes sense more. All right, let's go to the next question, guys. The diagram shows some biological molecules. Consider which biological molecule forms a polymer with structural role in plants. Yeah, so which one forms structural role in plants? Pause the video and try it. Structural role. Now let's name it. A is what? Okay, A is alpha glucose. How do I know? Because the OH attached to this first carbon atom, this OH is below the plane. This is the plane of the ring. Now this is the ring structure. Can you see? That's ring structure. The plane is straight line, this horizontal line from here. Okay. So the OH attached to this is the first carbon atom. The OH attached to this first carbon atom is below the plane. Okay, let me show you. I think I should do it well. So look at, if we draw a plane structure, like this. Okay. Yeah, of course you can see just horizontal line across. Don't forget that this is carbon 1, this is carbon 2, and the difference is just in carbon 1. This is carbon 3, right? That's carbon 4, right? For the 2 monomers of glucose, alpha, and that is carbon 6. And okay, so if you check here, can you see if you draw the plane of the rings, okay, so this is also carbon 1, this is carbon 2, all right, this is carbon 3, all right, that's carbon 4, that's carbon 5, and that's 6, all right. And you see we're interested in this OH group. Do you see? Attached to the first carbon atom. This is OH group attached to the carbon atom, all right, to the first carbon atom here, right? So what do we say, guys? Right? Which one a structural role in plant? That's cellulose. Okay. So which one is a monomer of cellulose? Right. This is alpha glucose because the OH attached to the first carbon atom is below the plane of the ring structure. So that's the monomer of starch and glycogen. They are not structural. They don't play a structural role. Okay. So A cannot be the answer. Did you get? Now, of course, if you come to this is glycerol. Okay, 1 2 3 carboxyl group, right? That's glycerol. it doesn't play structural role in plants. Okay. Now if you check, this is a pentose sugar. This is a nitrogenous base. Okay, so this is just adenosine. There's no phosphate group here. It doesn't play structural role in plants. But if you check this is beta glucose, because OH attached to the first carbon atom is above the plane of ring structure. So beta glucose is a monomer of cellulose, and cellulose plays structural role, okay, in plants. So the answer is delta D. Okay, so let's go, guys. Which molecule is a disaccharide and reducing sugar? Di to monosaccharides. Okay, pause the video and try it. Which of them is a disaccharide and it's a reducing sugar? Pause it. You can see fructose is a monosaccharide. Okay, glucose monosaccharide. These two are disaccharide, maltose and sucrose. But sucrose is non-reducing sugar. Maltose is reducing sugar. So it's C. Okay, so maltose is disaccharide and reducing sugar. Let's go to the next question. The diagram shows bonding in part of a polysaccharide. Okay, there's a polysaccharide. Can you see this? This is very cute. Fine, man. Which type of glycosidic bond is shown at position X? Can you see X? Which type of bond is shown there? So these are your options. I want you to choose the option for yourself. Which ones? Okay. Now, don't forget that this position X, that if you check this is carbon 1 here, right? Carbon 2 is here, okay? Carbon 3 is here, this junction, okay? Carbon 4, all right? This is carbon 5, all right? This one here is carbon 5. Then this is carbon 6, okay? So that means this is also carbon 1 of that one. Okay, this is carbon 2 of this molecule, right? So this bond, basically, people, they are formed between carbon 1 and carbon 6. Alright, and if you check, it's branched. So beta glucose does not form branch structures, alpha. So this is going to be alpha 1 6, right? So it's not going to be alpha 1 4 is between one carbon, alright, the first carbon atom of a molecule of glucose and the fourth carbon atom of another adjacent molecule, right? But this is the first carbon atom here and the 6. So this is 1 6, alright? It's not beta, it's alpha. Okay, let's go to the next question, guys. Which statement about triglycerides and phospholipids are correct? Both of them are lipids. Okay, so which is correct? I want you to pause the video and fry this chicken. Fry this fish for me. I'm hungry, guys. Alright, let's look at number 1. Okay, number 1 said fatty acids in triglycerides may be saturated or unsaturated, but in phospholipid they are always saturated. Well, this is not correct. It's the same thing. Alright, it may be saturated or unsaturated, alright, in phospholipid, just like in triglycerides. So this is not correct. Okay, number 2 says triglycerides, consider triglycerides and phospholipids both have hydrophobic region. Yes, both of them have hydrophobic region. That's the fatty acid part of them. Alright, fatty acid tail. Number 3, guys, if this is triglyceride, this is, alright, just to sketch it. Now, triglyceride means 3 fatty acid. This is the first, second, this is the third. Okay. Now this is phospholipid, right? Okay, it has 2, right, 2 tails. Okay, if I should get into the game again with this thing, you can see this is triglyceride and phospholipid both have hydrophobic region. So their tails are the hydrophobic region. So they have tails, and these have 3 fatty acid, 2 fatty acid tail for this is phospholipid, this triglyceride. All right, this has 3. Now, number 3 says triglyceride are non-polar molecules. Phospholipid are polar. Well, molecules. So if you check, yes, this is non-polar. Yes, but phospholipid has a polar region. All right, this is the head. This is the phosphate group and the non-polar region. Okay, so And of course, there's no option that has only 2 as the correct answer. So 2 and 3 are the most likely answer we have here. Okay, so we're going to choose 3. So the option that has 2 and 3 should be the correct answer. That's C. All right. So ask me a question, guys. Let's go to the next question. 4 extracts from different plant materials were made and tested with Benedict solution. Okay, the first one, the extract were boiled with Benedict solution for 240 seconds, and the final solution was recorded. Okay, so this is extracts. This is color produced after 240. So number 1, red. Okay, and they were boiled with Benedict solution. Okay, so pause the video. All right, they say which sequence of plant extracts represent an increasing quantity of reducing sugar. So pause the video and try. Which sequence? All right, remember that Benedict solution. But however, you gotta pause this video and try by yourself. Remember that Benedict solution is used to test for reducing sugar, guys. And if reducing sugar is present, if it's not present, it will be blue. But if it is present, okay, like we normally say, guys, yeah, if it is not present, all right, the color, all right, will remain blue. All right, but if it is present, it can change to green, right? Yeah, so green is present but in small quantity. It can change to yellow. Yellow is, okay, higher quantity, right? Is that okay? Yeah, it can change to maybe orange. Orange is also moderate, okay? Quantity based on their concentration. And then if it is in high concentration, to be red. Okay, so red is brick red. You can call it brick red. Brick red shows the highest concentration. So if this means blue means no concentration, that's 0. Okay, green means little. Okay, so they say increasing quantity, so it's going to increase from blue to green, from 0 to small quantity to moderate quantity, which is yellow and orange, and to the highest quantity, which is red. Okay, so it's going to move from blue. Blue should be the first, right? It should be the first. The second should be green, right? Blue is 3. The third should be yellow, and the last should be red. All right, so which option starts with blue? Blue is 3. All right, okay, C and D. The next is green. Green is 4. Okay, so this option makes sense. What do you think? Yep. So let's go to the next question, guys. Yeah, is which have properties that are dependent on hydrogen bonds? Cellulose, a molecule of hemoglobin water. Okay, so try them out. Cellulose, yes, because cellulose, pause the video and try it. Cellulose molecules are linked by hydrogen bonds, okay, to form microfibril. Right, so hydrogen bonds are here. Molecule of hemoglobin, yes, hemoglobin is a protein, so hydrogen bonds are formed is part of the interactions between R groups to form secondary structure, okay, and tertiary structure. Yes, so water molecules have hydrogen bonds. It causes water molecules to bind to each other. That's cohesion. So all of them have it. Okay, they all have it. All right, let's go to the next question. Which statement is correct? Cellulose, glycogen, look at that. Which of them is correct? Now, A says cellulose, glycogen, and amylopectin are all polymers. Now, pause the video and try it. They are all polymers. Yeah, they are all polymers. Yes, cellulose is a polymer of beta glucose, alpha glucose, alpha glucose. They are polymers. But why is the other ones wrong? Ribose, amylase, and phospholipid are all macromolecules. No. Phospholipid, of course, ribose is a reducing sugar. Is a monosaccharide. 5 carbon sugar. So it's not a macromolecule. It's not a large molecule. Macro means large. Macro, large. Starch, glucose, and amylose are all monomers. No, starch is not a monomer. The sucrose, the oxalose, and amylopectin are all polysaccharides. Nope. Sucrose is disaccharide, not polysaccharide. Deoxyribose is a monosaccharide. So the accurate answer there is option A. Alright, the diagram shows two amino acids. Some of the hydrogen atoms are numbered 1 to 6. Okay. Alright, which two numbered hydrogen atoms could contribute to the production of a molecule of water when a peptide bond forms between these two molecules? Okay, so what do you think is the answer, guys? Pause the video and try it. Let's see what I got. Okay, exam is on 11th of June, okay, 2024. Are you ready for this exam, guys? Paper 1 is an important component. Okay, now look at this, guys. Yeah, so during the formation of peptide bond is formed between, okay, the terminal carboxylic group and the terminal amine group. So these two should form it. 3 and 4. Is there any option that has 3 and 4? Nope. Now 3 and 6 can't form it because they are all carboxylic group. All right. Now let's check this one. What a 1? This is amine. This is a terminal amine group, right? Is at the end. Terminal 1. This is a terminal carboxylic group. Okay. What about 1 and 6? Yes. So B is the answer. All right. Peptide bond is formed between terminal amine group and terminal carboxyl group, right? Okay, so either this and this or that and that. Okay, our student wrote 4 statements about water. Number 1 says water has high specific heat capacity, which maintains temperature of water within cells. All right, mammals rely on water having a relatively low latent heat of oxidation to keep them cool. 3. When a negatively charged ion is added to water, the positive charge on the hydrogen atom is attracted to the ions. When surrounded by water, non-polar molecules tend to be pushed apart from one another. Okay. So, which statements are correct? Okay. Pause this video and try it. Which statements are correct? So, number 1 says water has high specific heat capacity, which maintains the temperature of water within cells. Yes, this is correct. This is high specific heat capacity, right? Causes water, okay, to change sluggishly as temperature. The temperature of water changes slowly, so water plays a hemostatic role. Okay, this is correct. Now number 2 says mammals rely on water having a relatively low latent heat of oxygen. Water actually doesn't have low latent heat of oxygen, they have high. So this is wrong. So number 1 is correct. So we are dealing with A and B. Okay. Number 2 is wrong. Yes, A and B is saying wrong. Number 3 says when a negatively charged ion is added to water, the positively charged on hydrogen are positive and negative are attracted to each other. So 3 is correct. So A and B are also saying the same thing. When surrounded by water, non-polar molecules tend to be pushed apart from one another. Which statements are correct? Okay, now non-polar molecules are attracted to each other when they're surrounded by water. That's called hydrophobic interaction. Non-polar molecules are attracted to each other. They are not pushed away from each other because they have a common enemy. Their common enemy is water. They say that people with same common enemy can unite. So they don't fight each other. They have a common enemy called water. They don't like water. They are hydrophobic, water hating. They literally hate water. They want to kill water, So they're supposed to unite, not to fight each other. So, which one is correct? I think it's A. What do you think, guys? Now, this says number 4 is wrong. Okay, it should be B. Right? They push apart from each other. So 4 is not correct. I think so. Okay. So, see, 4 is not correct. The answer is B. Okay. Which bonds are present in all viruses? Do you like viruses? COVID is caused by a virus. Alright, now pause the video and fry this chicken. I'm hungry. Now say which bonds are present in all viruses? Now phosphodiester bonds, pause the video and try it. Phosphodiester bonds are formed between nucleotides. So they are found in DNA or RNA because both DNA and RNA are nucleic acid. And DNA or RNA are found in all viruses. Some viruses have DNA, some have RNA. Okay, so this is correct. Okay, peptide bond. Yeah, viruses have protein coat called capsid. So proteins have peptide bond. Okay, all right, and these bonds, especially peptide bond, they could also be called covalent bond. Okay, protein also have disulfide is a covalent bond. Right, so all the three bonds are correct. The answer is A. A student carried out Benedict tests on 4 different concentrations of glucose solution and then recorded time taken for the first appearance of color change. That's the end point. The student found it difficult to identify, okay, because of maybe eye problem, the first appearance of a color, and consistently, now see, consistently is very important, timed each solution for 2 seconds after the color change, right, first appeared. This is very important. Okay, consistently after the first color change, the color change first appeared. This introduces a source of error in the experiment. Okay, let's go. Which statement about this error are correct? Yeah, so look at these statements. Which ones are correct? Pause the video. Let's see what you can do, people. Yeah, so let's see what the ones that are correct. Which statements about this error are correct? Okay, let's go into it. Number 1, the effect of the error will be reduced if the student performs 3 repeats at each concentration glucose. No, this doesn't take care of that accuracy. All right, so this is error on accuracy. Okay, so this takes care of validity, repeats, and calculating the mean. Deals with validity, not accuracy. Okay, so this isn't correct. I don't think it's correct. Okay, now number 2 says the error will prevent students from identifying which solution has the highest concentration. No, it's easy. It's just that the students cannot identify the actual time. It's always 2 seconds after the color change. Okay, the highest concentration of glucose will always be brick red. Okay, brick red is different from yellow or orange or green. So the student will identify it. So this statement says the error will prevent the student from identifying. This is not correct. Okay, number 3 says the error is, okay, now so let's take it out. Okay, so I don't think this is correct. This is wrong. Okay, I don't think this is correct also. Number 3 says the error is systematic as the students consistently timed each solution for 2 seconds. Yeah, this is correct. Okay, so the answer is delta, 3 only. All right, which statement, let me get my pointer. Come on, laser pointer. Which statements about amylopectin and glycogen are correct? Amylopectin and glycogen. Okay, these are the statements, guys. So pause the video and tell me the answer, and I'll send you a pack of chocolate. All right, which statement about amylopectin and glycogen are correct? Amylopectin and glycogen contains 1,4-glycoside bond. Yes, they have it. Okay, amylopectin contains beta glucose. Nope, beta glucose is not a monomer of amylopectin. It's a monomer of cellulose. So this is correct. That is wrong. 3 says glycogen contains more 1,6 branches than amylopectin. That's correct because glycogen is highly branched, small branch. So it's 1 and 3. Okay, yeah, let's go on. How many statements can see how many statements about fatty acids are correct? Okay, check it out. Yeah, how many statements are correct? Let's see how many statements are correct. Number 1 says fatty acids are either saturated or unsaturated. Yes, that's correct. Okay, yeah, depending on the number of double bond. If they have double bond, they're saturated. No double bond, saturated. Okay, fatty acid always have at least 2 double bonds. It's not always at least 2 double bonds. Okay, it depends on the type of fatty acids. 3 says long chain of fatty acid are very good in its story. Yes, because they have a lot of hydrogen atoms. Hydrogen atoms are cellular fuel. Okay, so 3 is correct, 1 is correct. Okay, let's check 4. Fatty acids are insoluble in water. Yeah, that's correct. And do not affect the water potential of the cell. Yeah, that's correct. Because they are insoluble in water, they will not affect the water potential of water. So number 1 is correct, 2 is wrong, okay, 3 long chains is correct, 4 absolutely correct. So how many statements are correct? Okay, 3 of them. C. The diagram represents an amino acid. Okay. All right. See that amino acid. So R represents a variable side chain. What is not a possible side chain there? Good. This is R group. Yeah. Pause the video and try it. Which one out of these 4 is not possible here? Okay. What is not a? All right. So you can check this R here. Okay. This is a methane or methyl group. Okay. Yeah, it's possible. This is also possible. But if you check D, you see that D is talking about glycerol. Can you see glycerol? There's OH in the first carbon atom, OH in the second carbon. Okay. Glycerol is part of lipid, not part of amino acid. Is that okay? So D is likely, all right, not a possible side chain because this is part of lipid. Okay. All right. Which polymers are present in all viruses and all prokaryotes? We solved this question in our cell structure, but we can solve it if you have watched that video. All right. All viruses. Pause the video. You should be able to tell me the answer. Okay. All viruses have nucleic acid, DNA or RNA. And DNA and RNA, they are all polynucleotides. So 1 is there. All viruses have proteins, all right, capsid, protein code. So that's there. Not all viruses have polysaccharides. So it's 1 and 2. Okay? So 1 and 2 is the answer. 1 and 2. So this is there. That is there. That's not always there. Okay? All right, 1 and 2. Which set of steps is the best method? Okay, the best method for conducting the emulsion test for lipids. Yes, look at them. All right. Okay, which is the best method? Okay, look at it. Pause the video and try it. Which one is the best method? Okay, A says add 2 cm cube of water to the sample. No, you don't add it to the sample. Okay, because it's not soluble. Lipids are not soluble, so you don't add it. Don't start. B says add 2 cm cube of ethanol to the sample and shake. Yeah, that's correct. You're getting it. Okay, let's go on. Pour the ethanol into a test tube containing water. That's good. And boil. No, it don't boil. It don't boil, so this is out. C says add 2 cm cube of water to the sample and shake. No, this is wrong. You don't add water to the test solution, the sample, because it contains lipids. Lipids are not soluble. So, okay, we are left with delta D, option D. Add 2 cm cube of ethanol to the sample and shake. That's correct. Pour the ethanol into a test tube containing 2 cm cube of water and shake again. Lipids are present if the mixture becomes cloudy. Yeah, that's exactly, absolutely correct. So D is it. So a student was provided with a solution of carbohydrate. They removed 2 samples from the solution and performed tests on each sample as shown. All right. Which statement explains this result? Okay, look at it. This is carbohydrate solution. Pause the video. I'd like you to try this. Okay, try it, and know how far you've got. Okay, so try it out. So you say which statement explains the result. Remember, this is carbohydrate solution, and they will have into sample 1 and sample 2. So the sample 1, Benedict test, Benedict solution remain blue. So there's no reducing sugar there. Okay? Because if you remain solution remain blue, no solution reducing sugar. The sample 2, boiled with dilute hydrochloric acid, neutralized with Benedict solution, tested. Okay, Benedict solution turned yellow. Yellow means this is test for non-reducing sugar. So it has no reducing sugar. Okay, let's see. Condensation reaction occurs in sample to release reducing sugar. Yeah, there's no condensation reaction. Condensation reaction joins the reducing sugar. It does not release them. Okay? Glycosyl bonds in a polysaccharide have been broken to release reducing sugar. Yeah, this makes sense. So when you boiled here, we broke the bond and we release reducing sugar. That was why we got yellow. All right, but let's check. See, sample 1 shows that sucrose is present in the carbohydrate solution. All right, sample 1 shows that sucrose is present. Okay, now sample 1 is used. This is a test for reducing sugar. It's not a test for non-reducing sugar. Sucrose is non-reducing, so this is test for sucrose. All right, this is test for reducing sugar. This is for non-reducing sugar. Reducing sugar, non-reducing sugar. Sucrose is non-reducing sugar, so that's not correct. This is the change in color to a yellow solution shows that glucose is present. Well, it doesn't show that glucose is present. It shows that sucrose is present. Okay, that's test for sucrose, or it shows that sucrose is present. All right, reducing sugar. All right, released by sucrose. All right, so what do you think is the answer? Process B. All right. So when molecules contain at least, at least 2 double bonds, which one contains? I think that's the typo. 2 double bonds. Okay. Pause the video. All right. So sucrose doesn't have double bonds at all. So anything that has to do with sucrose is out of it. So we're left with this. Simple. So collagen, hydrogen bond. It has double bond because it consists of amino acid. Amino acids have double bond. Hemoglobin the same. So the answer is B. All right. What describes cellulose? Cellulose. Pause the video and get the answer. A branch chain. Okay. Pause the video now and solve it. Cellulose is on branch, so A and B out. Let's come to C. C is an unreactive linear chain. Okay, it's a linear chain, but the monomer is not alpha. The monomer is beta. So that takes us to D. D is the answer. Okay. All right. Which part of the structure of hemoglobin carries oxygen? Pause the video and try it, guys. Pause the video. 4 polypeptide chains, they are not the where the oxygen binds. Okay. The hem groups, here is the hem groups. Hydrogen bonds, no hydrogen. So the hem groups are the binding sites, all right, of oxygen in hemoglobin. Okay, they're the binding site. That's B. All right. What is the maximum number of hydrogen bonds that can be formed between 2 single water molecules? All right. So what is maximum water? Okay, I want to try it. So what is the maximum water hydrogen bond that can be formed? Remember that water molecules stick to each other. So if this is a water molecule, for example, all right, water molecules stick to each other. This is hydrogen, all right, it's partially positively charged. Okay, that's positive. Okay, I hope you have solved this. This is negative, right? So can you see water? This is water molecules, all right? So this water molecule can form hydrogen bond with another water molecule here. Okay, watch, this is another water molecule here. It has, let's assume, of course, the hydrogen, the oxygen is there. This hydrogen, all right. Okay, this is hydrogen. Is that okay? Yeah. So this is partially positively charged. So this forms hydrogen bond with this. Okay. Now, see, guys. Water molecules form hydrogen bond with this one. Okay. It uses this to form hydrogen bond with the next water molecule. Okay. All right. Or uses this to form with the next water molecule. It cannot form 2 hydrogen bonds with just 1 water molecule. So the maximum is 1. This one. Okay. So that it can bond with other water molecules. This is because that allows water molecules to form a column, especially for water transport in xylem. All right, you should be able to attach to other water molecules. All right, like 2. For example, if this is a water molecule, let's assume that this is a water molecule here. There's another water molecule here. There's another water molecule here. Water molecule, and this is inside xylem wall. Okay, sorry, xylem. Okay, so water molecules forming a column. So you see water molecules, they are forming unbroken column. So water molecules should be able to form hydrogen bond with each other. So this forms hydrogen bond with this. It forms with this. Also, can you see? It forms hydrogen bond with this. It forms hydrogen bond with this. It can also form hydrogen bond, all right, with the wall of xylem due to adhesion. Okay, you can see, so that the water column is maintained. So they form hydrogen bonds with cellulose or hydrophilic part of lignin on the xylem wall. You can see each of them. So you see, the maximum it should form, all right, hydrogen bond should form with another water molecule is one. So it can only form one between this and this, just one, this one. Between this and this, just this one. So I think the answer is A. All right, thank you guys for hanging out. All right, so I'm wishing you the best in your exam. So I'm going to be dealing with enzymes. The next video is on enzymes topicals. All right, see you. Don't forget, this is your teacher, your tutor. All right, Shedro, and this is 13 Universal. See you next. Bye.